Hello student, once again welcome to Onus classes. Today in my video I am going to discuss some important question of the chapter biotechnology, which is one of the important unit of for your coming exam. So from the biotechnology chapter generally questions comes from recombinant DNA technology and question also come from PCR. PCR is a part of the recombinant DNA technology, then SCP and then electrophoresis. Other one more question also comes, suppose what is the application of biotechnology, what is biotechnology, like that. But mainly these three topics are very much important from this unit, means from this chapter. So if you go through the recombinant DNA technologies thoroughly, then many questions will come from the technology. So first comes the question, that is define single cell protein, means SCP. Write this advantage, name some common microbes as SCP producer. So this from this question has come in 2014 and 2015. So in 12 board exam you will not get direct copy paste question. You will means I can give you the important conception or important topic, so any question can come from that topic. So first is the definition of SCP. It is a protein obtained from a single cell organism used at food supplement. So if one more question comes, just write up to here. Single cell protein is obtained from the dried cells of microbes such as bacteria, yeast, algae, and fungi. It provides instant energy. If 2 marks come, then you write up to here. If 1 marks come, you just write up to here. Then what is its advantage? So advantage are these three advantage. It has high rate of multiplication, large biomass can be produced, it helps in reduce the pollutant, it is not affected by the weather condition. And example, here I am giving you three examples. Generally question come or give one example of means SCP. So one example of bacteria, one of yeast, then one of algae I have given you. Next, define gel electrophoresis. Mention two application of this technology. So gel electrophoresis is a laboratory method used to separate the mixture of DNA, RNA or protein according to the molecular size. Means DNA and RNA all have different molecular size. So by this process, they are separated according to their size only. In gel electrophoresis, the molecule to be separated are pushed by an electric field through a gel that contains small pores. And what is the application? Application are, it is used in DNA analysis and the study of DNA fragment used in separation of biological molecules. Third question, what is the principle of biotechnology? So main principle is what? Selection of desired gene, selection of vector of the transfer of gene known as cloning, insertion of recombinant DNA into the host, and maintaining the introduced DNA into the host so that it pass on the next generation. And what? So if you forgotten 4 point, at least first 3 point you don't forget. First 3 point you have to remember. Then next question, what is the recombinant DNA technology? Write about the process of recombinant DNA technology. So this is a long question. If this question come, it will carry 5 marks. But if you go through the whole answer of this question, many short question will come from this. Means from this question, within the answer of this question, lots of answer are giving which type of question may come. So first definition of recombinant DNA technology refers to the joining together of DNA molecule from two different species that are inserted into a host organism to produce new genetic combination that add a value of science, medicine, agriculture, and industry. It means by this process, two different DNA from two different organism bring and join together and a genetically modified DNA is made, and which is introduced into a host. As a result, the genetically modified new organism is produced. Now come for the different steps of or process of DNA technology. First, isolation of the genetic material. The first step of rDNA, recombinant DNA technology, short form it is known as rDNA, is to isolate the desired DNA in its pure form. Since DNA exists within the cell membrane along with other macromolecules such as RNA, polysaccharides, protein and lipids, it must be separated, purified with involved enzymes such as lysosome, cellulose, chitinase, ribonuclease, protease, etc. Excuse me. Next step is, so first step, DNA is isolated from other molecules, means from RNA, protein, just DNA is separated out. Second is restriction enzyme. Restriction enzyme means it acts as a molecular scissor, means which is used to cut the DNA. And these are known as restriction enzyme digestion process. This is known as restriction. So one more question may come, what is restriction enzyme digestion? Then just you have to write the process of cutting the DNA molecule into different segments is known as restriction enzyme digestion. Next, amplification using PCR. So amplification means multiply. So polymer chain reaction, full form of PCR is polymer chain reaction, is a method making the multiple copy of DNA sequence using the enzyme DNA polymerase. So by using, so one more question come, which enzyme help in the amplification of DNA molecule? You have to write DNA polymerase, means it help to multiply the number of DNA molecule fragment. It helps to amplify a single copy of or a few copy of DNA into thousands or millions of copy. PCR reaction has the following components. So short question may come to mark. What are the different component of PCR? Then you have to write this. Template, DNA to be amplified, primer, small chemical synthesis, oligonucleotide that are complementary to a region of DNA. Enzyme means DNA polymerase, then nucleotide needed to extend the primer by the enzyme. Next step, lignation of the DNA molecule. So the purified DNA and a vector of interest are cut with the same restriction enzyme and joined together using the enzyme DNA ligase is lignation. The restriction DNA is a the resulting DNA is a recombinant DNA. Means suppose one extracted DNA molecule plus vector DNA both are jointed together, and as a result what happened? As a result the recombinant DNA is produced where the desired DNA portions are used. So this is the diagram. So this is the modified plastic. This is the diagram of modified plastics. Insertion of the recombinant DNA into the host. In this step, the recombinant DNA, after forming this desired recombinant DNA, this is the, after forming the recombinant DNA where the gene of interest means required gene is present, it is introduced into a recipient host cell. This process is known as transformation. For one mark, this term is important. Obtaining the foreign gene product, the recombinant DNA multiply in the host and is expressed as a protein under optimal condition, and this is now called the recombinant protein. So recombinant protein definition means the recombinant DNA when multiplied within the host is expressed itself as a protein which is known as recombinant protein. Last step is downstream processing. Before protein is marketed as a final product, it is subjected to downstream processing the includes separation and purification, formulation with suitable preservation, clinical trial or test efficiency and safety of the product, and quality control test. Next question, write the application of recombinant DNA technology. It is came in 2017. Recombinant DNA is widely used in biotechnology, medicine and research. The most common application is in the basic research, in which technology is important to most current work biological and biochemical sciences. So here different application I have given you total 3 applications. Generally 2 marks are come, so then you just write 2 application. How does enzyme endonuclease help in the formation of recombinant DNA? So endonuclease means worked as a molecular seizure. So recombinant DNA is formed by using the restriction enzyme that cut the double stranded at a particular point. First it cut the DNA fragment in a particular point. The same enzyme is used to cut the second piece of DNA also. With this, when the fragments are mixed together, the complementary strand of each other strand will bind with those of other, forming a recombinant DNA molecule. What are the raw material used in Miller experiment? So ammonia, methane, hydrogen and water vapor. Write the principle of PCR technique, which is a part of recombinant DNA technology. Already I have explained previously, still I am giving you the answer here. Mention the role of PCR molecule diagnosis. PCR means polymerase chain reaction, based on using the ability of DNA polymerase to synthesize new strand of DNA complementary to the other template strand. In PCR, a short segment of DNA is amplified using primer mediated enzyme DNA polymerase synthesis new strand of DNA complementary to the template DNA. The DNA polymerase can add a nucleotide to the pre-existing 3OH group only. How amplification of DNA is done by PCR technique. It is also a part of recombinant DNA technology. Means from PCR technique, lots of question has come in many years. So it is very much important topic for your coming exam. The steps of PCR, the key ingredients of PCR techniques are tag polymerase, primer, template DNA, and nucleotide. So for 1 mark, this may become what are the key ingredient of PCR. Then you just write tag polymerase, primer, template DNA, and nucleotide. The ingredients are assembled in a tube along with the cofactor needed for by the enzyme and are put through the repeated cycle heating and cooling and allow the DNA to be synthesized. The basic steps are: first one, denaturation. First you just remember the name of the steps, then you remember the steps. So from these steps, sometimes what happens, just a question are given that write the denaturation step of PCR technique, then you just write this point only, denaturation. And it is go through in 96 degrees centigrade temperature, is heat the reaction strongly to separate or denature the DNA strand. By this means at this temperature the DNA strands are separated. This provides the single stranded template for the next step. Next, annealing. So it is cooled down and the temperature come to 55 to 65 degrees centigrade, and at the temperature the primer can bind to the complementary sequence on the single stranded template DNA. Next, extension. Again the reaction is heated to 72 degrees centigrade and as a result tag polymerase extend the primer, synthesizing new DNA strands. So this is the diagram. Look, first this is the normal DNA strand. Denaturation means heat the DNA. Then both the strands are separated from each other. Next, primer annealing 55 degrees centigrade. Here what happened? This sorry look, the primer can bind a complementary, so in this case the primer are bind with a complementary DNA strand, then again it is heated to 72 degrees centigrade, then tag polymers help the primer to extend towards both the complementary strand, and as a result, so this is the primer, it is extend like this, and in this extend like this, and finally two daughter DNA strand are formed. So this cycle is repeated 25 to 35 times in a typical PCR reaction, which generally takes 2 to 4 hours depending on the length of the DNA region being copied. So how many times generally required? 2 to 4 hours, and how many cycles? 25 to 35 times. And finally, so from as a result, from one DNA, lots of DNA strands are formed. Next, full form of PCR, polymerase chain reaction, already I have repeated. What is tag polymerase? DNA replication in an organism. PCR requires a DNA polymerase enzyme that make new strand of DNA using existing strand as template. And DNA polymerase means DNA. What is DNA polymerase? It is the enzyme that make new strand of DNA and using the old existing strand. And this DNA polymerase used in PCR is known as tag polymerase. So what is tag polymerase? Means the DNA polymerase enzyme that is used in PCR technique is known as tag polymerase. What is PCR primer? Like other DNA polymerase, tag polymerase can only make DNA if it is given a primer, a short sequence of nucleotide that provide a starting point for DNA synthesis. Means the the sequence of nucleotide that give the starting point for DNA synthesis is known as PCR primer. So primer means what? It is the sequence of nucleotide that provide the starting point for DNA synthesis. What is molecular diagnosis? Molecular diagnosis is done to identify a particular sequence of nucleotide in a mixture of nucleotide. Next, how DNA is isolated in purified from a bacterial cell. The steps are: the cell is broken to release the DNA molecules along with other macromolecules such as RNA, protein, polysaccharide, and also lipids. This is done by treating the bacterial cell with the enzyme lysosome. Then RNA is removed by treatment with protease. Protein is removed by treatment with protease. Then ethanol is added to the precipitation of DNA. The purified DNA can be collected in the form of fine thread in the suspension. Next, what is the principle of cell electrophoresis? So this is repeated in 3 years. So 2014, 15 and 17 also. I have forgotten to write the year here. Gel electrophoresis is a technique used in the laboratory to separate the source molecule like DNA, RNA and proteins according to their size. And what are the principle? Charged molecules move through a cell when electric current is passed across it, and electric current is applied across the cell so that one end of the cell has positive charge and the other end has a negative charge. The movement of charged molecule is called migration, and molecule migrate towards the opposite charge. So this is the principle, or this is the process also you can write. What are the steps of cell electrophoresis? Only if 1 mark come, you just write the name, or 2 marks also. You, if 2 marks come, you just write the step and then you write the answer of question number 11 also. So steps are pouring the gel, preparing your sample, loading the gel, running the gel, and finally staining the gel. Write the application of gel electrophoresis. So visualize band of a molecular marker to genotype individual plants, verify amplification by PCR or sequence reaction. Check the quality and quantity of the genomic DNA after DNA extraction and separate DNA fragments to clone as a specific band. Next, short notes has come on gene cloning. Gene cloning means the formation of exact DNA or gene copy using genetic engineering technique. The DNA is extracted from an organism and the gene is copied and a clone is formed. It is one of the methods used for the isolation and amplification of specific gene. How insulin is produced through genetic engineering? There are total 6 steps. Human insulin is extracted from pancreas cell and an insulin producing gene is isolated. Plasmid DNA is extracted from a bacterium and cut with a restriction enzyme forming plasmid vector. Insert a human insulin producing gene into the bacterial plasmid vector to form the recombinant DNA of human insulin producing gene. Introducing the recombinant DNA into the bacterial cell to form a recombinant bacterium. The recombinant bacterium multiply in the fermentation tank and produce human insulin. Insulin is extracted, purified, and bottled, and this is then ready to be injected to the diabetic patient. What do you mean by GEAC? How it controls the genetic engineering research? GEAC stands for Genetic Engineering Approval Committee. The committee have to set certain rules and guidelines to approve of GMO, means genetically modified organism. It has the power to approve the or reject use of genetically modified organism at large scale for research. It also regulates the use, storage, import, export, and hazard microbes or genetically modified cell and organism in India. What is GMO? What is genetically modified crop? What is GMO do to food? Genetically modified organisms are those in which DNA from a desired organism is inserted in the laboratory. They are known as transgenic organism also. So what is transgenic organism? Then you have to write the same thing. Or if a question come, what is transgenic animal? Then you have to write the animal where the DNA is inserted from a desired organism. The plant in which DNA has been modified using genetic engineering method for agriculture purpose are called genetically modified crop or GM crop. Example is BT brinjal, GM mustard, etc. Then GMO help to protect the crop by providing the resistance to a specific disease or a insect, ensuring greater food production. GMO are also important for the important source of medicine. Then last question is explain the gene therapy by citing the example of ADA deficiency. Gene therapy is a replacement of a defective or faulty gene with a normal or healthy gene to correct the genetic disorder. Adenosine deaminase deficiency is caused due to the deletion of ADA gene coding this enzyme. To correct this disorder, lymphocytes are isolated from the blood or bone marrow of the patient and grow in the vitro culture. The defaulty ADA gene is removed and in the situ hybridization of the normal ADA gene is done through the transformation technique using the adenoviral vector. The transformed lymphocytes are subsequently transferred to the patient under aseptic conditions. So these are the important questions which are very much important from the biotechnology chapter for your coming exam. If you go through all these questions, any type of question come, you can answer the questions. Okay, then for the next video, till then, thank you and goodbye.